a glimpse at the frightening decline of California's coastal fisheries, the ethical challenges of genomics, predicting and managing the effects of global climate change on California's water supply, research in predicting and preventing heart disease, all on this edition of On Beyond.
La Jolla Cove, 1941. A group of high school kids, 4 of us to be exact, started the second oldest diving club in the country called the Manta Ray Club. All these cliffs here were covered with abalone. All the cracks in the rocks had abalone in them. On the minus tide, people came out and gathered them. Same was true of lobster. We found them in the tide pools here. The California black sea bass used to swim around the edges of the kelp bed or through the kelp beds. I've personally seen them here the size of Volkswagens. I've seen them up to, at least 300 pounds here. White sea bass were the same way. They'd come in schools.
The kelp forest. It may appear to be thriving at the surface, but a different reality lies below. Like so many other ecosystems today, kelp forests have seen a dramatic decline in animal size and abundance. A number of species are ghosts, no longer filling their former ecological roles. Why have so many species virtually disappeared from the kelp forest? We know some answers.
Kelp forests themselves are massively impacted by the oceanographic climate. The oceanographic climate includes El Niños, in which the nutrient-poor water starves the kelp plants, to massive storms, which might remove almost 100% of the kelp forest in one storm. So there's a great deal of natural variability in the kelp forest.
And this variability affects fish. But after decades of study, we now know that giant kelp rebounds quickly when conditions are favorable. Yet much of the animal life has only declined. Why? Scientific evidence suggests we have harvested too many animals too fast for too long. To combat declines, we've regulated fishing through limits and bans on specific species. And in coastal waters, we have even outlawed the use of gill nets, a particularly destructive form of fishing that took a tremendous toll on kelp forest species like the white sea bass. Protection has helped reverse the decline of some depleted populations. Divers are beginning to see giant sea bass once again in kelp forests around the Channel Islands off California. Yet despite the protection of individual species, and the efforts of fishermen to comply with regulations, we still have ghost forests. How can we help restore the kelp forest and other essential ocean ecosystems? New research supports adjusting the way we manage fisheries. Scientific studies show the value of creating networks of marine reserves, carefully selected areas of the ocean in which all species and their habitat are completely protected from harvesting of any kind. In existing no-take reserves, fish abundance nearly doubled in as little as 2 to 4 years. The size of fish increased by almost 1/3, and 3 times more offspring were produced. As sea life increases in protected areas, it naturally spills over to adjacent areas where fishing can take place. Not all people support this course of action. For some, it may spell a loss of income or changes in time-honored traditions. But experts believe there will be far greater long-term costs if we continue to manage individual species while ignoring the ecosystems that sustain them. Less than 1% of the ocean is now protected in no-take reserves, and few such reserves exist in U.S. waters. La Jolla's ecological reserve is a rare exception. The future of the kelp forest lies in our hands, but creation of marine reserves will not happen without strong public support. What will our legacy be? We can settle for telling future generations about the ghosts of kelp forest past. Or we can choose to take an active role to restore the riches of this valuable ecosystem.
Professor Beer, Ethan, if I may, thank you for joining us tonight. You are a developmental biologist. Can you give us a brief description of your work and the kind of work that means that you do in your lab?
Well, developmental biologists, ever since Aristotle, have been interested in the basic problem of how you go from something as simple as an egg, without any apparent structure inside of it, to a very complex organism like a fly, a human, or a plant. How does that happen? It's something I think people have puzzled about for a long time, and it's in a certain sense it's a question of who we are and where did we come from.
As well as being a biologist and an active researcher, you've become an author and authored a book called The Coiled Spring. What is the meaning of the title, and how did you come upon this idea for the title?
Well, the coiled spring is meant to represent DNA, which is the genetic material that controls the developmental processes and other processes, all other processes ultimately in living organisms. And DNA is a helical molecule, and so the idea was that the coiled spring is the sort of repository of the information which is launched forward when an egg gets fertilized. And it's depicted, I think, very nicely here on the cover, which was drawn by Judy Kadihi, my editor. What we see is the DNA within the egg springing forward from the egg and launching the developmental programs of these diverse organisms.
What was your motivation for writing the book?
In the last 20 years, there has been enormous progress in the field of developmental biology that's taken it from what was primarily a descriptive science to a science where we now understand things in mechanistic terms. And we know, not everything, of course, about how development works, but we have a very good outline from how you go from making an egg to making a differentiated structure. Another thing that has happened at the same time that we've begun to understand in mechanistic terms things like development and other aspects of biology too, we are now being presented with the full genomic sequences of many different organisms, including humans and the model genetic organisms like, say, fruit flies. And the combination of these two sort of breakthroughs is creating a revolution, which I think in many ways is equivalent to the Industrial Revolution of the 19th century or the computer technology revolution of the 20th century, and I think it will be remembered as such as history goes on. And I think because of that, it's a very important matter for people who are thinking about all kinds of social issues to consider the implications of what we know about development and other mechanistic aspects about biology in terms of the consequences that this will have on a variety of different technological and social levels.
Well, help us understand some of those things that are bringing biology into the social arena, because the picture of biology for us non-biologists has changed radically in the last 30 years, 20, 10, even over the last year. So could you bring us up to speed on some of the major changes in understanding it, especially in terms of developmental biology?
Well, there's sort of three major points that I try to make in the book. The first is a question of, does development happen all in one sort of large step, which there used to be what was called the homunculus model of development that sort of considered the idea that an embryo is really just a miniature full-grown creature and that during development it just gets bigger. And the alternative to that, which is actually what we believe and is argued in the book, is that actually instead of development happening by just sort of capitalizing on information that's already present in highly detailed structural format in the early embryo, actually the embryo, the embryo, the egg that is laid by the mother, has relatively modest amounts of information in it. And based on that modest amount of information, some simple steps take place upon fertilization that end up subdividing the embryo into a few domains, for example, along the nose to tail axis into the head, trunk, and tail, so relatively modest subdivisions of the body plan. And then the genes that are acting in those different three basic domains interact with one another to create yet another round of information that breaks things up into stripes that correspond ultimately to the various segments of the animal. So what we believe is that development goes on as a series of many simple steps rather than as one very big step all at once. The second point, which also has some practical consequences, is that within the animal kingdom, invertebrate organisms such as the fruit fly, which is what we study in the lab, and vertebrates such as mice, frogs, ourselves, share a remarkable number of similarities in the early stages of their development. So, for example, the way you make the subdivisions along the nose to tail axis are almost, the mechanisms for that are almost exactly the same. For example, the segment in a fly that would have wings on it, as the same gene that's specifying that domain of the body in a fly defines the same relative trunk-like position in a human being or in a mouse. And in fact, you could take a vertebrate gene and put it into the fly, and it would function in the same way that the fly gene does to make a fly structure. And so what this has allowed us to do, these similarities, as well as similarities, for example, in how the heart forms, how the muscle forms, how the nervous system forms, how eyes probably form, how outgrowths that are appendage-like are initiated, has allowed us to create an image of what our most recent common ancestor must have been. That is the common ancestor of say, a fly and a human. And contrary to the view that prevailed when I was a student, which was that that was a very primitive organism, we now have an image of it as being a rather evolved, complex organism, and that explains why it is that humans and flies share so much in common. We derive from a highly complex common ancestor, and those mechanisms, those genetic mechanisms for creating that common ancestor have simply not been lost during the course of evolution. And then the third point, I guess I try to make in the book, is that the ancestor of plants and animals was a very simple creature, perhaps a single-cell type organism. And yet, we find that there are remarkable similarities in the general strategies, not in this case the details of the exact genes that carry out a certain function, but the general strategies for how one makes certain types of structures. For example, the flat spatula-like wing structure of a fly versus the flat corresponding leaf-like structures of plants. It turns out that in both cases, extremely similar genetic mechanisms are at play, and these have come as great surprises, and yet we're finding many, many examples of these kinds of similarities, suggesting that actually there aren't a million ways to do it. There really are a few general strategies that evolution has independently worked on during the course of the evolution of these two great experiments in multicellular complex life.
Probably the most interesting and relevant thing is what this understanding of the basic genetic characteristics of organisms means.
What it means, in part, is that there are many, many genes present in something like a model genetic organism like a fruit fly and a human that function the same way. And not only function the same way, but you can actually take the gene from a vertebrate or even the human and put it into a fly, and it will do what the fly's own gene would do normally, so that they're functionally equivalent. And that means that you can then take genes from something like a human condition, a disease, and try to understand what the basis for that disease would be by studying it in this model genetic system. And so this is a remarkable, opportunity to go in there and take the human gene, the normal human gene, the gene that's causing the disease condition, and compare their activities in a fly. And we've already done a little bit of that in the lab, and others have too. And so far, all the indications are is that this should be a very successful way of beginning to understand how genes that cause human disease work. And then, of course, we can then go back once we've understood what it is that the gene is doing, and perhaps learn a little bit about how to mitigate the circumstances that arise from the disease form of it, and then go back to humans and ultimately down the road develop therapies that help treat those diseases. So I think there are incredibly profound implications for human health.
What are we getting into? Where does this put humanity?
It's a great challenge. And that's actually also one of the reasons for writing the book, is I think it's important that people understand the terms of this knowledge, what it means, what we can do with it, so that we can all figure out together how we should use it. There obviously are incredibly wonderful things that could be done with it for the betterment of humankind. And these range, again, from some of the health matters that I've been discussing, curing things like cancer, the recent success, for example, in curing bubble babies by gene therapy, and the ability to screen for myriads of genetic disorders. And then in the agricultural sector, which in fact is the arena where the impact, I think, will be felt first and is being felt first, there are remarkable opportunities to address some very, very important problems that are critical on a global scale, like how are we going to feed a world population that is predicted to double within the next 100 years. There's no way we can boost up the food production based on current methods to accommodate that increase in world population.
Now, there has been a great hue and cry about organisms like this, genetically modified organisms. There's great debate going on in Europe, and it's coming across the United States, as well as eugenics, the idea of controlling human genetics. In some cases, it's reasonable, but in a lot of cases, and one could say most cases, it hasn't been reasonable. Could you give us some examples of the benefits versus the alternatives in this case, in the case of pesticides?
I think the Bt example is a good one. Again, this is a toxin that's made in bacteria. It's a protein. It's a protein that vertebrates, mammals, can eat in large, tablespoonfuls without suffering any adverse effects of any kind. Yet, it causes little crystals to form in the guts of insects that ultimately lead to the death of the insect pest. Now, one of the things that can be done, again, with transgenic or genetically modified GMO plants, is to put the genes that make these bacteria, that encode these bacterial proteins, within the plant itself, so that the plant makes the protein, you don't have to spray it on, and then the plant is resistant to the insect pest. Now, in some cases, in fact, in a way that somewhat puzzles me, to be honest with you, organic farmers and others, I think, are taking a slightly misguided point of view, which is to worry about the potential problems that could arise from this disproportionately to the reality of it, and not appreciate some of the great benefits. You have to balance what is the good of using, say, something like the Bt toxin versus the alternative, because there's always got to be an alternative. And the alternative, at least on the scale that agriculture is being conducted today, is the reliance on very harsh chemical pesticides.
Dealing with the human organism is more complicated, both in terms of technology and in ethics, but the benefits could be dramatically compelling. And you have an example in your book which speaks to how dramatic and how compelling this could be.
There's been a recent case, and there are many other genetically based diseases. It could equally well be any of those others, but that of Marfan syndrome, the condition that afflicted President Abraham Lincoln. And there have been two people who are known to be carriers for this disease. In a typical disease of this kind, the parents are carriers, and 1/4 of their offspring, in principle, could be mutant, that is, have 2 bad copies of the gene, and they would suffer from the disease, unlike their parents who are just carriers. Half of the progeny would be expected to be carriers, and a quarter of them wouldn't get either bad copy of the gene, and so they would be completely free of the disease. That is, they wouldn't be affected by the disease, nor would they be carriers. So this couple, wishing not to certainly have offspring that had the disease, decided to go the in vitro route. And what they did is they had 8 embryos fertilized in vitro. They grew them up to a point where a few cells could be taken from each one to be tested genetically for whether they had or not these mutant forms of the gene. And what they found is that 3 of the 8, slightly more than one would expect by chance alone, which would be 2 out of 8, 1/4, were completely free of the disease. That is, they didn't have either bad copy of the gene. So those 3 embryos were implanted into the mother, 2 of which took, and the mother gave birth to 2 children that now not only don't have Marfan syndrome, they don't even carry it. Well, that's the age of eugenics. That's the ability to change the genetic makeup of your offspring in a way where even a silent trait, like being a carrier, has been erased.
Now, in terms of the word eugenics, to me, that, I'm sorry, that's positive eugenics. You know, there have been the horrific Frankenstein nightmarish visions of eugenics, but that portends something very powerful and very compelling for human health.
You know, at the level of the story that I told you today, I think most people would agree that no harm has been done. All you've done is take a disease that the parents certainly didn't want to transmit to their kids and just relieve their kids of the burden of having to worry about that themselves in the next generation. But when you start talking about 5,000 traits, and then as genetic information progresses and our understanding as it is rapidly accelerating progresses into the understanding of other traits, like, for example, you know, tendency for substance abuse, and then just strength, intelligence, and all kinds of other properties of humans that are known to at least have a significant genetic component. At some point, I think anybody is going to start getting nervous, right? Because now we really are talking about the bad kind of eugenics again. And where's that line? And how do you think about it? And where do you, you know, how do you draw it? And I think that's what I really hope that people start thinking about, because this is the time to think about it before it's already a fait accompli. That we now have an opportunity to say, all right, you know, and I'm not purporting to have any of the solutions here, but I think it's everybody needs to think about this, and we as a whole culture, nation, and planet need to come to terms, to grips with what we can do in the future, and everybody has to be part of that decision. I think that's why it's really important for people to become informed, is that we need informed people to agree to where we're going to be drawing that line, because it's a very gray line on a very serious landscape.
In a very complex world.
In a very complex world, and it has to be drawn, because the not taking a decision is just as much of taking a decision as making the wrong decision.
In a sense, Pandora has opened the box, the genie's out of the bottle,
so to speak.
It's out. And in every other case in history, when the technology, something's developed, it's been used for good or not for good. And as a biologist, you have a certain knowledge and insight that some of us don't share. And your book does go a long way to giving people a very good, thorough, fundamental understanding of the processes that we have to understand that are facing us in the future. But I also know that you're a parent, and we all know what that means, that it's the dearest thing to you. Perhaps you can best communicate your motivation for the book by sharing for us what you would like to see the wise use of this technology bring to the world of your children and your grandchildren.
I think my general answer to that would be, again, harnessing, making the most of the power of the technology. I mean, again, if we make the analogy to the Industrial Revolution, sure, there were some really bad things that came from the Industrial Revolution before the pollution controls were in place. You know, there were excesses that came from that end of it. But on the other hand, it is also what gave us the standard of living that we enjoy in the developed world. And I think the the opportunity exists with this new technical revolution, the biotechnological revolution, that I think that the expansion of these technologies and the implementation of them in a wise way will make the world a better place for the next generation, my son, grandson, grandchildren. I think the challenge we must all rise to is to make sure that the good, which is clearly there as a potential to come from these technologies, is realized with as little negative consequences possible from having taken some unavoidable necessary risks along the way.
Climate and water have a very intimate relationship, and it's an interesting one in a lot of different ways, but one of the most intriguing things about it is that water is the medium that climate is expressed in. It moves across the earth in different forms. It moves from solids to liquids to gases, and it's the medium of exchange for all the energy that is stored on the earth, at least at the surface of the earth and in the oceans, and that's driven primarily by solar radiation. And that energy is absorbed to a large degree by the water on the earth and moved around the earth by the rotation of the earth and by what we think of as weather patterns. And so broad-scale changes in climate that we think of, particularly in the case of climate warming, that affect the distribution of water, are really changes in the distribution of energy and the way the energy is stored and moved on the earth by water in its various forms. When we say liquid, we're talking both about fresh and salt water, so we're talking not only about the water in the oceans, which is of course salt, but also the water that's stored in glaciers that we have in our rivers and streams, and the bulk of our fresh water is stored in glaciers, and that's why issues having to do with the change in the distribution and condition of glaciers, both at the North and South Poles and in the mountains all over the earth, are of concern. So one of the most interesting things about this is that climate's everywhere. Everywhere on Earth there's climate. There's also weather, of course, but the weather is kind of a localized or a more localized expression of climate. Climate is a long-term pattern of energy distribution on the Earth, and the location and variation, spatial variation and temporal variation, we see in different parts of the Earth as localized weather. We have to talk about different scales of measurement when we talk about climate in order for it to make sense, because we go all the way from global scales, which is where climate modeling and climate scientists tend to think most completely about the problem. But then in order for it to have meaning for society and for people in general, it's important to make that transition of information down to the local decision-making processes, the political decisions, the social decisions that we need to make in order to ensure stability of our human population, our human societies, and our ability to continue to survive and thrive on the earth as an ecosystem with us embedded in it. So when we look at these different scales, water crosses different spatial scales, and we talk about global scales, regional scales, and local scales. And the global scales are, as you might imagine, the entire Earth's surface. And we talk about regional. Regional is a fuzzier kind of concept. Regional can mean things like the western continental United States, could mean the Western Hemisphere. So regional really is contextually dependent, but local scales tend to mean things on the order of city scales, the scales where political decisions are made, or you are working in a bay or an estuary in your neighborhood and you can see pollution that's having an impact there, or changes in the distribution of salt and fresh water that affect the life and your ability to enjoy that, or for the organisms that live there to thrive well or not. And the interesting thing about climate, water, and scale is that we go across all of these scales. We go from global to regional to local scales, and water is important on all of those different scales, but it's the things that we concern ourselves with at each scale varies, and that's something we want to pay attention to. Climate has the power to change where water comes from and where it goes. And that's really the theme that we're trying to address here. How does climate change drive the distribution of water in a way that affects human societies all the way from global to regional to local scales.
The global change problem, of course, is one that has come into sharper focus, and I would say, fortunately, that the state of California and the California legislature is starting to think about the prospects of what might happen to the California water system with the prospect of climate change, which is a very real prospect. Using present-day models, there's a pretty strong likelihood that we'll see increases in temperature of something like 2 to perhaps 4 degrees Celsius above today's levels. Now, an important aspect of California's water system is the natural storage that is afforded to us from the California snowpack. The snowpack acts as a natural reservoir. California's artificial reservoir system is only large enough to capture about a year's worth of flow. So the amount of storage that we get in the snowpack is a really critical aspect of our water plan. That is, the ability to count on a spring snowmelt that is occurring during, when you think about it, a pretty manageable part of the year. In other words, it's happening after the real active winter storms have occurred, so that that water can be essentially managed without worrying about flood protection in the California reservoir system. The reservoirs we have are not only water storage vehicles, but they're shields for floods, and consequently, during the wintertime, we have to allow for a safety measure in those systems. So we can't catch all the water. But once the spring is upon us and the winter storm track has receded to the north, then we can let those levels creep up, so we can catch that water with more confidence that we're not going to have a safety issue. The problem with global change is that, first of all, we're not going to have as much snow, and secondly, the snow that we get will likely melt faster. So that using a fairly state-of-the-art model and I would say a conservative estimate of temperature change for the state, that is only about 2 degrees Celsius, our guess is that we're going to lose about half of the present day of snow accumulation in the combined Sierra drainages by the end of the present century, that is by about 2090 or so. So this is going to be an aspect of climate change that, assuming these models are fairly on the mark, we're going to have to contend with, and it's going to be a big challenge for California water, because we're going to have a lot of water coming off earlier than what we have today. And in fact, with the reservoir system that we have, it's pretty clear that we don't have enough storage capacity to hold it all. So we're going to have to adopt some new strategies.
At the Hydrologic Research Center, in collaboration with the Georgia Water Resources Institute, we developed a software package to assist reservoir managers in their job. And this is a software system that we call INFORM, for Integrated Forecast and Reservoir Management. It has components to take climate forecasts, downscale them down to the watershed sizes as appropriate, generate inflow forecasts for the reservoirs in a variety of forms, including their uncertainty. And lastly, take these forecasts and at a given risk level, generate trade-offs among the various uses of the reservoir water, such as hydroelectric power production, flood control, and downstream uses, domestic, agricultural, and others. Uncertainties are very important for INFORM. One of the objectives of the system is to try to characterize all the sources of uncertainty and incorporate these characterizations to the trade-off curves that the manager will see, so that the risk level that we provide is representative of these uncertainties. These uncertainties start from the uncertainties in the climate forecasts, which may be substantial for a given region on the globe, to the uncertainties of going from the large scale of the climate forecast to the small scale of the upstream catchment, to the uncertainties of the hydrologic models in terms of topographic, soil, land use, land cover data, to the uncertainty of the stream channels in terms of how exactly they convey water, how much sediment exists that constricts the conveyance, and other such issues. As part of this experimentation with INFORM, we also looked at the future. We looked at the next 30 years of climate as simulated with state-of-the-art climate models. And the results we found showed that under projected weather scenarios for California, the use of such integrated forecast management system, in fact, is much more beneficial than at the present conditions. The reason for that is that under weather conditions, even the large reservoirs such as Folsom will be very close to capacity. So it would matter much more to make judicious decisions to be able to manage essentially a relatively smaller volume that you have available for the reservoir. This is the wasteful spill of water downstream. 3 different scenarios of managing the reservoir as shown. The current practices, they don't use the forecast. The middle part is the integrated system that uses the forecasting management, the climate forecast and the hydrology forecast. And the last one is the system in a case where the manager is clairvoyant, knows precisely what's going to happen. And it's the ideal. I mean, you can never know that. And so we simply put it there to say that with present-day technology and models, we're approaching that.
We know that there are challenges to both policy and technology. The technology challenges because we were trying to come up with new ways of measuring and analyzing these systems and new ways of using computer and information technologies to address them. There are also challenges from environmental policy because without environmental information, the policy making is basically guesswork and very subjective. And technology, on the other hand, without a particular application, is pretty much an academic exercise. So we have an opportunity with water to make a transition across the policies and technology areas in a way that satisfies not only scientific curiosity purposes, but can very much enhance the ability of our society to address these challenges by better information, more timely information, and improve our ability to coexist with the natural systems that we live in. We have data, we combine with models, and the challenge is to construct those models in such a way that that data can be accurately reflected and reflect the natural processes that we're trying to model. We can then drive the models with observed weather, for example, precipitation, wind, and other factors that are present and important in the natural system, and take those results and present them as maps or present them visually in other forms and compare these with observations. And it's the process of doing predictions and comparison with observations that helps us understand what's going on. And once we have confidence in the modeling capabilities, then we want to use these in a predictive mode to make forecasts that would help us make decisions, intelligent decisions about how we should behave as a society to change our infrastructure, because if we do this, it will cause that result. Or if we do something different, we will have perhaps a more beneficial result. So the challenge and the excitement from a computer science point of view is to take the technologies, the big computers that we have, the large numbers of processors, and the ability to take large quantities of data, integrate them in a systematic way, and then present them in a useful visual presentation. That's one of the exciting things about using computers in this kind of a setting.
Dr. Tsimikas, thanks for speaking with us today.
Thank you for inviting me.
I understand that you're currently developing a non-invasive technique to image arterial plaque buildup. Can you tell us a little bit more about that?
Yes, we have recently cited some research into developing a novel technique, a non-invasive technique that will identify atherosclerotic lesions. These are the lesions that cause plaque buildup within arteries that cause heart attacks. And this will be a technique that's going to use an antibody that will be labeled and then will be injected, and it will be a nuclear, most likely, technique that will non-invasively detect blockages in certain arteries of interest.
I see. Before we go into detail, could you explain why we want to detect this condition early on?
There's very clear data that most patients that have heart attacks, or at least 50% of them, have no previous warning signs. And this makes a test that's non-invasive very imperative to detect these patients before they have all the complications of a heart attack. The current techniques will only detect blockages when patients have symptoms, because that's when the blockages become severe enough that they can be detected. For example, when a patient has symptoms, usually they have an over 70% stenosis in the coronary arteries.
Now, what exactly is stenosis?
A stenosis is a obstruction, a 70% obstruction in the lumen of the vessel wall that would obstruct the blood flow to the heart muscle. So that before anybody has symptoms, they need to have at least that amount of blockage. Now, the current tests all rely on having a abnormality in blood flow. For example, a treadmill stress test will only be abnormal if there's a specific blockage that's over 70%, for example. Same thing with a nuclear scan or a stress echocardiogram. And certainly an angiogram, if you do that, will show you earlier blockages, but most patients don't have symptoms before that, so you wouldn't necessarily perform that test. And it's also an invasive test. So that what we need is a test that's non-invasive, that has very low risk, that will detect plaques that are less than 70%, because we know now that most heart attacks are not caused by very severe blockages. Severe blockages will cause angina and give people symptoms, and they will have time to report to their doctor and get an evaluation. Whereas heart attacks usually are caused very suddenly by a certain process called plaque rupture and thrombosis, which involves blood clots forming over a very small plaque in the artery, and then in a sudden onset of the heart attack.
Could you describe the actual process of a heart attack?
If this is your normal blood vessel, a blood vessel that will have a plaque in it will look like this. Now, this is the beginning of the plaque. Now, if this plaque does not have more than 70% stenosis, you will not be able to detect it by any of the current standard tests. They will all be negative because the blood flow through that vessel is completely normal. Now, what happens when you have a heart attack in most cases is that this plaque that's not obstructive has erosion or cracks in it. And those cracks allow blood to flow into it. And when blood flows into it, a blood clot forms suddenly. And this is what causes a heart attack, because now no blood can travel down into the heart muscle. And this is usually an abrupt onset. And this happens over 1 million times every year in the United States. And about 300,000 of these people die suddenly before they can get help. So our goal with this test is to develop a way to image that plaque that there's no other way to detect at this point, non-invasively.
And how does this imaging technique actually work?
This technique involves a new way to image atherosclerotic lesions. And these are the plaques that build up within arteries. The plaques have deposits of abnormal cholesterol, and we call that oxidized LDL. And this oxidized LDL is what makes the plaque grow and enlarge. And there's large deposits of this oxidized LDL within these plaques. And what we're doing is we're targeting that oxidized LDL by what is a monoclonal antibody to oxidized LDL. Now, what this is, it's a protein that was developed in a mouse that binds very strongly to oxidized LDL, but not normal cholesterol. The LDL is low density lipoprotein. That's the abnormal cholesterol, the bad cholesterol that people know very well about and trying to lower it to prevent heart attacks. So this antibody, this protein, will only bind to abnormal cholesterol deposits. So the idea is, and this was developed in Dr. Joe Whitsom's laboratory at UCSD And my role came when I went to train with him in atherosclerosis, and then we tested this technique. So the idea was born in his lab and subsequently tested in his lab. And the idea is that we take this antibody and we put a radioactive tracer called technetium, which is a very common imaging agent that we normally do now for nuclear scans. And this tracer will light up, if you can think of it that way, light up the antibody. So we inject the antibody into a subject. We allow it to then accumulate where there's abnormal cholesterol. Now that antibody now will give a signal. It will not bind to normal arteries. It will only bind to where the deposits are. And it emits the signal that we put on there. And once we allow it to accumulate in a certain amount, we then will come in with a camera, just like a typical CAT scan or that sort of camera. And as the signal emits from the patient, we can detect it where on the artery the signal is. So by knowing where the signal is coming from, we can then say that that patient will have cholesterol deposits in that artery. And those are the cholesterol deposits that are associated with all the adverse effects of atherosclerosis, such as a heart attack, for example. Current techniques for detecting blockages are treadmill test, which involves looking at EKG abnormalities. There's nuclear scans, which detect blood flow. There's stress echocardiography, which detects abnormal heart motion. And you see all these are indirect measurements. Now these are all non-invasive tests, meaning that you don't have to go into the arteries to get the information. The problem with all those tests is they're not 100% accurate. At best, they're about 80% accurate in giving you the right information. So sometimes they're wrong in either way. They can say that you have something when you don't, and vice versa. Now the gold standard is coronary angiography, which involves putting a catheter into the artery and inserting that into the heart and then injecting dye directly into the artery and getting an image. That technique is invasive. It has some small risks, but it's an excellent test for defining where the blockages are. The problem with that test is it does not predict who will have a heart attack because it's not the severe blockages that cause them, it's the minor ones. And a lot of those patients wouldn't necessarily have an angiogram. There's also the issue of cost and the complications of the procedure, which makes it a test that you really only want to do when there's something to fix and the patient needs something repaired. So it's not really a screening test. And this test that we're trying to develop would be more of a screening test for people that are very high risk, that have high cholesterol level or diabetes or high blood pressure or smoke, that we can do in an outpatient setting. It would be minimal risk to the patient, but it would give us the information where the plaques are, and perhaps, and of course we would have to prove this, perhaps prognosticate and maybe make us, give us information about who is a high-risk patient so that we can do more aggressive interventions, like more aggressive lipid-lowering therapy, weight loss, dietary modifications, and medications.
What needs to be done to begin the human study?
This technique is relatively new, and we really only started working with it in 1995 and on. And we have validated in principle that this will work in patients, but we do not have any patient data yet. We've done lots of animal studies, and what we've shown is that when we inject the antibody that's labeled, it identifies these plaques and that it identifies them quantitatively, meaning that the more plaque you have, the more uptake of this agent you have, which is a very nice thing, so that you can actually have a sense of how much plaque is there. And we've also shown that you can detect depletion of this bad cholesterol from the vessel wall.
Could you show us some images from these studies?
This is a mouse aorta, and a mouse aorta is very small. It's about 5 centimeters long, quite tiny. Now, what you're looking at here is the entire aorta. Now, the aorta is the largest blood vessel in the body, and that goes from the heart all the way down to the legs. Now, mice have very small coronary arteries, so we can't image those. Obviously, the human arteries are much larger. But this is an example of what kind of imaging we could do. This is a mouse aorta that has plaques. This mouse has high cholesterol level, and you see here the pinkish red material here is all the beginning of blockages. You see there's very specific areas that it accumulates in. Now, these are the kind of plaques that not necessarily cause an obstruction, but if you use the human analogy, these are the kind of plaques that would potentially cause a heart attack. Now, we want to be able to detect these particular plaques in patients. And we first obviously have to validate it in animals. Now, this is a mouse that was injected with a small amount of this antibody that was labeled with a tracer that we can detect. And what you're seeing here is that after the antibody accumulated in the plaque, we get a picture of that in this picture here. And you can see here, if you compare these two images, the dark here represents the accumulation of this antibody with the signal in it. And you can see it correlates perfectly. It's almost a mirror image of where the plaque is, so that when you have plaque, you have signal. We have no plaque in the gray area here. This is the normal part. You have no signal. To show you how sensitive this technique is, you can see here this tiny little red spot represents a small plaque, and this is less than 1 millimeter. And you notice here on the, this is called an autoradiograph, that little plaque lights up when you do the imaging. Now, this is not a gamma camera image. This is an X-ray image, which is a little bit different. This is not a scan. But you see the point this makes is that even tiny little plaques will be detected by this technique. Now, to show you another example, this is a rabbit aorta, which is much larger. And this is about the actual size. And the aorta is opened up, and this is why it looks like a Y. This is the part that comes out of the heart, and the rest of it that goes down into the legs. This animal has much more atherosclerosis. You see here, this whole aorta is caked over with atherosclerosis, and then you have some smaller lesions. Again, if you look to see where the antibody accumulates, it only accumulates where there's plaque. And notice there's little gaps here. There's no plaque. There's no antibody signal. So this technique looks very nice for detecting only where the plaques accumulate and not the normal vessel, which would be very nice so that when we inject it, ultimately in patients, we will only detect, the signal will only be coming from the plaques. Let me now give you an example of what an actual image looks like with the camera. This is an alive, in this case happens to be a rabbit. And the rabbit was injected with this antibody that was labeled with a tracer, and it was allowed to accumulate, and then an image was obtained. Now, let me show you what it looks like in a rabbit that does not have any blockages. You can see the aorta here in this rabbit has no blockages in it. And when we do an image of it at a certain amount of time, that image should, if there was plaque there, would light up the middle part of the body. Now, these are the kidneys, and this is the liver, and this is the bladder. And those also light up because the tracer naturally accumulates there. Anything you inject in a patient will go everywhere in the body, so that these also will give you signal, but we're not interested in that anyways. We're only interested in the aorta, which is in the middle of this particular animal. Now you see there's no signal in the middle where you would expect the aorta to be. So because this rabbit has no plaque, we get no signal. Now let me show you another example where we're actually detecting the signal, again, in a live rabbit. And you see here, this rabbit has lots of plaques all through its aorta. And you see now the middle part is all lighting up. And what we're detecting here is these plaque deposits. So when we ultimately do this in patients, we will actually just focus in on the area we want to image and detect whether there's a plaque or not.
What are the foreseen advantages of this procedure?
The advantage of this procedure is that, one, it will be non-invasive, which is very nice. Now, there's two other techniques now that are non-invasive tests besides the usual stress tests that I measured. One of them is, and this is now coming into the public arena, is there's something called ultra-fast CT scans, which actually detect calcium deposits in arteries. And now they're being used as a marker of atherosclerosis. Now, calcium tends to be associated more with stable plaques that don't necessarily cause heart attacks. So the usefulness of that technique needs still to be determined, but it may not predict as well about who will have a heart attack. It may tell you if there's a beginning of a blockage, but it may not predict. What I'm hoping this technique will ultimately prove is that because we're now imaging the lipid deposits, which are associated with heart attacks, that it will predict heart attacks, and it may be a better technique. Of course, that has to be validated in patients. So the advantage will be it'll be non-invasive. It may be applicable with other techniques like magnetic resonance imaging, nuclear, or even ultrasound, which we're evaluating. And it may allow us to predict, although this has to be proven, whether the patients will have heart attacks or not. And until we do human imaging, we won't know that. But the potential is very good for being a technique that will predict who will have heart attacks. And at this point, that technique does not exist. None of our tests truly predict who will have a heart attack. They predict who will have a blockage, but not necessarily a heart attack. The other advantage is you will be able to follow patients over time with it, and because it allows you to detect changes in the plaques, you may be able to very strongly tell a patient whether they're getting worse or better and allow more specific intervention.
So this could actually be an early screening process.
The way I envision it is this will be the kind of technique that will be used in patients that have several risk factors that are asymptomatic, and that you will be able to diagnose deposits of cholesterol that may allow you to have some predictive value. It will be used in patients that are already on lipid-lowering medications that you want to see if their plaques are changing for the better. It will also be useful in animal models, so we can test these agents, new agents, in the laboratory before we give them to patients to see if we actually can detect changes in these lesions. And that'll be a nice research technique to perhaps test some of these agents before they're used in patients.
Where do you see this project heading in the future?
Well, this technique, if it is as good as we hope it will be, will have some advantages. We know that a lot of young patients die suddenly of heart disease. And you know, you hear the typical example, a patient has a treadmill test on Friday. He may be a 45-year-old executive, he has kids, maybe a little bit older, and he's told that there's nothing wrong with his heart. He may have had some minor symptoms before, maybe some risk factors for heart disease. And then on Monday, he drops dead of a heart attack. And this is the kind of patient that I'm trying to identify before they have their heart attack. And this technique hopefully will pick up the blockages so early that we can actually have time to correct them before this happens. And unfortunately, about 300,000 patients every year die suddenly. And over 90% of them usually have underlying obstructions and then develop an arrhythmia on top of that. And one of the advantages of this technique will hopefully be to detect those high-risk patients. Other advantages of that, it will be easier to perform, and hopefully that will allow better screening and more earlier intervention. I think the whole field of cardiology has gone from reactionary medicine, where you wait for the heart attack and you develop an ICU and you treat it, to developing great techniques to treat earlier problems now with angioplasty and bypass. But really the 21st century is going to have prevention as its goal. And it's hard to get people to, and everybody's human, to do something good for them unless you can identify a risk. If you identify a risk, the motivation then becomes very real to allow a change in their behavior to improve their outcome. So that's the way I see this technique, is that it will really allow early detection and motivate people and physicians and the whole healthcare system to intervene and prevent problems rather than treating the end stages of the disease.
Great. Thanks for sharing your time with us, and we wish you the greatest success with this work in the future.
Thank you very much.